we heard, I think, just a couple of really fantastic talks that cover, with just the two individuals we have here, a great spectrum from the beginnings of technologies for new measurements coming out, as Lars talked about, all the way through commercializing and hitting massive commercial opportunities, as Ravi talked about. And so maybe to get this started, I'll just throw a question to them. Those two worlds are obviously very important, and there's a divide between them in terms of how you go from the things that are really cutting edge in the lab to what goes commercially. And I'm curious about both of their perspectives on what's the optimal way to do those handoffs, and and what are really the bottlenecks in this field as we try to have personalized analytic devices revolutionize health, and what do you see as the big issues?
So one of the things we found out as I was working with a lot of universities, except for a few, very tough to get them to actually commercialize technology because they're in a different domain. So what I've encouraged now within our company is actually funded research projects where the focus is actually commercialization. So I actually fund projects will say, here's the problem I'm trying to solve. How do I go about using your research acumen and bring it to a commercial scale? So 3 of the things which we did came out of that. One of them is this. This actually started as a research project in a university a long time ago. The second one was this whole concept of how do you take an echo, which is a visual image, and then come up with diagnostics which are accurate. And so we funded actually a significant person in that field in echocardiography. So he was very focused on looking at 1 image every year, versus now he looks at 1 million images a year. And the idea is we gave him the tools to do that. And the third thing we did was work very closely with universities where we made it non-clinical. So for example, one of our devices increases circulation in the body. The question is, what performance increase do you want to see? For most athletes, it's, can I increase my VO2 capacity? And we funded that. So what we have done is try to cause the chasm by saying that both sides are equally important, because a lot of great research gets lost because it never gets commercialized. But if you can identify a specific market and a vertical, then we'll go commercialize it, we will fund it, you provide us research.
Great. Lars?
Yeah. I mean, you're exactly the type of people that we would go talk to if we're interested in commercialization. The Genome Center, I would say, is Stanford Genome Technology Center is a bit different than a standard academic lab. We have people coming in with engineering backgrounds, for example. It's a very interdisciplinary team, and so over the course of its history, we've founded about a company or spun out a company about almost 1 a year. That's either technology that was developed at the center, or it's an employee who went off and eventually founded a company. I think that that's different than what you would typically see in an academic lab. And the challenge is primarily getting enough funding in an academic setting in order to take a technology and actually show that it's going to be it's robust enough. So take a technology from proof of concept to the next stages. And I think that's often where in an academic setting we kind of like the technology falls into a gap or into a black hole and doesn't get developed further. So Silicon Valley, I think we benefit from being in that environment. And so my experience has been that VCs, et cetera, in the Valley are very eager to listen to ideas and to technologies. And so from my own experiences, like for example with the magnetic levitation, that's a technology where we're interested in exploring commercialization of it. And so we've had some initial meetings. It tends to be easy to get an initial meeting going, and then sometimes those pan out and sometimes they don't. Yeah.
Great, we have a question.
So I just wanted to ask a question and kind of leverage Mr. Sajwan's sense of global healthcare information and ask where you think these disruptive health technologies might first have a real impact, and is it the United States?
Actually, so we've actually had insurance companies come to us now and try to figure out how they could take our products and put that in their pipeline. A good example is, I won't name the company, but they gave us about 8,000 images and we ran them through our software on our system. And we were 99.2% accurate. And their best guess was 64% from their inside staff. So, and I think one of the cardiologists actually called me and says, like flipping a coin. Either you are right or I'm right, pick one. But so I think they realize that it's important, but you cannot also alienate a professional. Like the cardiologists are great, and the problem is they do a lot of work. So what we are focused on is not being the ones to replace a cardiologist, but assist them. So they focus on the abnormal, not on the normal stuff. So that message seems to resonate pretty well. And the other part of this whole thing from a cardiology standpoint, or even from a general practitioner standpoint, your trusted first individual you go to is your general practitioner. That's the first thing. So creating this channel of information, which is a GP can give you fairly good advice by looking at the data, rather than you having run it up and down the cardiology clinics. And that makes a big difference to the patient because they feel less stressed. And the white coat syndrome is much lesser in a doctor's office than it is in a hospital. So all those things add up to a better outcome. At least they're less depressed about what they're going through, rather than this whole thing. So we've seen different things. But I've seen insurance companies actually want to offer this as a standard procedure for an annual checkup. They will pay for it and actually drop the premiums if you do this every year. They know what the problem is, so they know how to address the cost. But if they don't know what the problem is, they don't know what the cost is. And that gives them that leverage to have that data. And it's at an individual level. It's not about geographies. It's about individuals.
Great. Another question? Three more questions. We'll take each.
So my question really goes to both of you. Lars, when you were talking about the experiments you were doing with your levitation by changing the environment with pH changes and then looking at the effect, this really relates also to, I think, the concepts that both of you are talking about, and that is steady state performance of systems versus under stress. And we can talk about what stress means in different environments. But I think that this field, when we move from diagnostics to prognostics, requires that you look at the fabric of reserve. And the fabric of reserve is tied into the dynamic of the delta under stress, right? And a lot of what we do in diagnosis, because we're in the pathognomonic side, is to look at a steady state of disease, which doesn't really give you much information interrogating reserve. There's a theme that goes through this whole conference, by the way, that I think relates to what I'm saying. By the way, I don't think that diagnosis of disease is a bad idea. I just think that there is a whole other side of this equation that relates to what are the parameters that we would use to start interrogating reserve in individuals that pertain to their accommodation. I guess we call it resilience. And what are the ways that we can use the technology they're developing today to really start to develop that level of understanding so we know how far away a person is on the resilience to a prognostic condition that might later become a diagnosis.
Right. Yeah, this is something we think about a lot at the molecular level. And I think you're touching upon something that relates, for example, to EQTL types of studies that are carried out where we're looking at the immediate downstream effects, let's say, of a genetic variant, like a disease variant in the genome. And in a lot of studies, people take disease tissue and then expression profile that or profile that with any omic technologies and get an association, try to get an association with a causal variant in the genome. But the problem is that you see a lot of consequences of being in the disease state if you look at individuals on the extremes. So like for an individual that would already have manifest the disease in an advanced state, it would be very difficult to identify the immediate causal players that lead to the disease in the first place, the early actors, and distinguish those from everything else that comes due to the system being in a stressed state. So at a molecular level, we tackle that by taking cells and exposing them to a variety of perturbations. And I think if you're thinking about a human disease, that could be taking a cell from a patient which has the genotype of interest and then exposing that to diverse stimuli. It could be generating diverse cell types, for example, from an iPS cell state, so that you could look at that genotype in the context of a lung, of a liver cell, of a muscle cell, et cetera, and carry out the omic characterizations across the board. My reasoning here is that if a genetic effect has an impact on a molecular pathway, then that impact on the pathway might be persistent across all the distinct environmental states that the cell is in, if it's immediately downstream of it. While all the other things which are a consequence of being in the disease state are not going to show up persistent across all the different environmental perturbations. If you think about it, so that's how I would approach this from a molecular standpoint and argue that we need to look at genotypes under a number of different perturbations. And I think that's also a large, there's a large gap in terms of what GWAS studies have done so far, where mainly we have variants currently. If you look at GWAS associations, variants annotated to be identified in a particular disease. But we know from sickle cell anemia, for example, that locus is detrimental for sickle cell, but it's beneficial for malaria. So what I'd like to see is a complete list of genetic variants in the human genome by a whole array of different environments that these variants have been tested on. And I think we're going to find a lot that variants are going to be detrimental in one condition, but beneficial in another. And that also relates to disease being a context of the environment that you're in. And that's what we try to do with drugs. We change the environment, keeping the genome constant, unless you're thinking about doing gene editing, like with CRISPR technologies. But if you're looking at a physiological, level, I think for patients, one of the ideas that we've been thinking about is to expose people to exercise stress. So exercise stress provides a physiological context where the entire system is perturbed and pushed to the extremes. So maybe we can unmask some subtle effects that show themselves at a molecular level. We can unmask those during stress because the effects might become larger during stress than they are in a completely resting state.
Great. Next question then.
So supposing somebody here develops a device prototype that has the potential to measure something that we can't measure very cheaply, that has the potential to inform us about health. What would you both say are the greatest obstacles from that prototype actually making a difference in the lives of people? And I mean kind of both from a scientific standpoint, but also socially and in the healthcare system. What would be the biggest obstacles, and also how can you, if you have suggestions on how to overcome them?
So we actually went through this about 3 months ago. What we found out that one of the biggest problems we saw was regulatory environment in different countries. US has FDA, Europe has CE, Singapore has HSA, so on and so forth. So there's a lot of different environments you got to deal with. And a lot of products actually get stuck or get lost in that phase because the cost to implement that is significant. You've got to get compliance, control groups, all kinds of things have to happen. So what we did, rather than waiting for someone else to fix the problem, we actually have our own inside FDA team. So we have a lot of people that we actually hired from FDA in our company. And we have people from HSA, we have people from CE, but they're all inside. And their goal is not to tell me what I cannot do, is to tell me what can I do. So take a product, and one example we have is this temperature sensor we just found, which is very unique. And this algorithm we found which actually replaces muscle biopsy by looking at glycogen levels to see using an echo. You can actually say whether the muscle is being repaired or not. So I can now offer this at a physiotherapist level or a massage therapist level because it's FDA predicated, proven, and the technology is there to drop the cost down dramatically. So we've found typically most products get stuck in the regulation stage. So you've got to empower the research team ahead of time and tell them what's lacking or what's not lacking. And you'll be shocked at how many design history files we have to go through just to go from US to Europe, actually. And so we have a team of 6 people doing just that. And so it's a lot of work. But once you know the process, it's streamlined very well. So my recommendation is if you have an idea, please call me anytime, no issues. We have a team which will get onto it. And we'll figure it out very quickly and get you to market. Yeah.
I have a question.
Oh.
If you look at any physiological function, we'll take cardiac function or something like that, just as a case. The medical profession has been spectacularly good at converting data into anxiety. And if you look at cardiac, let's say arrhythmias, a long QT interval or a type 1 AV block, these are on the margin of being something that you might actually be interested in, because a long QT might or might not indicate that you shouldn't take azithromycin, but that depends upon how sick you are. Since everybody who's not on the mean is off the mean, how do you avoid doing more harm than good by finding non-normal conditions and causing people to worry in a way that isn't actually actionable? And let me just emphasize that the medical profession is interested in profit opportunities, which means the bias is always to do something, even if the doing something requires generating a market, which means an anxious patient. So I guess I don't quite understand why more data is better.
I think we're going to see a change. My prediction would be over the next 20 years in how patients perceive, and how fast they worry about these types of data that we're generating with these technologies. Because my feeling is that we're going to be, as soon as we profile every healthy individual, that participates in a study like this, we're going to identify defects that they carry. And I think a good example is simply from the genome sequence already. Each of us carry, over a dozen loss of function mutations in the genome. Not all of them manifest into a phenotype because there's limited penetrance for many of these. But I think people will get more used to the fact that there's going to be information in the genome which is associated with certain probability of developing a disease, and understanding through the education system already what it means, what a probability means of developing a disease and a risk associated with a disease. So my hope is that through these longitudinal profiling technologies and technologies like you presented, which will allow you to monitor how you're doing, if you're progressing on a course of action towards disease or whether you're actually still in a healthy state, is going to help people. And I could see them embracing technologies like that, because if you carry a risk factor to develop, let's say, a cardiac disease, then you can be very interested in monitoring technology that will be able to monitor you longitudinally over time, so that you could be rest assured that you're still, everything's still good, the heart's still functioning, ECG looks normal, etc.
Do you think that PSA was a step forward or backwards?
I think, PSA and technologies to measure biomarkers in blood, I think, is a step forward. And I think we need more biomarkers to measure the health state of diverse organs from blood. A lot of that's going to be in the research setting initially.